Today, I'm going to discuss one part of the obstacles you may face when developing AI models for the clinic, which is kind of very early on this notion of always having positive and negative controls when you do any kind of analysis. And AI is no different. AI is very, very sophisticated and complex. And in a way, those controls are even more important. And I'm going to use radiomics as an example. Um, those are my disclosure, but none of them are relevant for today's talk. What I would like to let you with is to leave you with is really this concept of AI-driven segmentation in radiomics to enable the development of non-invasive imaging biomarkers, and also how to use some of the software that we developed in the lab to hopefully develop radiomics biomarkers that are a bit more robust. And so radiomics has been used, radiology images have been used for detection and diagnosis for quite a while. It's also been used for treatment planning for radiation therapy, and very importantly as well, monitoring response in clinical trials. So measuring the tumor size is the way we assess whether a drug is actually beneficial for the patient or not. And radiology is this very rich data modality where basically the image is a matrix of numbers that could have very different meanings. And for a human looking at the image, we interpret it in a certain way, but for a computer, there might be very subtle signals that could be extracted from those images. And so images are really not just images; they're really deep, complex data. Usually you start with the detection of a lesion in the patient, so a tumor that you segment into the 3D, and then you basically extract a lot of features computationally to kind of try to go beyond what the human eyes can detect. And there are many features related to shape, the intensity of the voxels, the way they arrange as texture, some intrinsic signal as wavelet from the images. And then from that, you basically kind of process this complex, very rich kind of voxel cube into a matrix of features, the what we call radiomics features. And again, it can be used for prognosis, try to predict patient survival or the aggressiveness of the disease, um, to detect the disease, to diagnose the disease, to stage, to plan the treatment. And radiomics, I mean, as you may imagine, with the advance of AI and GPU hardware, the number of publications on radiomics is really exploding. And one of the seminal publications that I've been really fortunate to be part of by Hugo Art is this paper really that was the first to look at large amount of radiological data and using computationally derived features on a large scale, so hundreds or thousands or even thousands of features to develop prognostic models. This study has many, many limitations that have been shown over the years, but it was nonetheless a bit of a seminal paper in the field. Um, then they basically, it went from a bit of each lab developing its own code to a bit of a maturity in the field with the publication of multiple packages to compute those radiomics features, and those packages were made open source. One example is PyRadiomics from the same lab from Hugo Art. That was kind of, I think, the first open source radiomics package computing like hundreds or I know more than 2,000 features. So that was kind of another kind of big event in the field is to have more available software that way. Um, And that really triggered a lot of activity in terms of software development. You can see here a series of packages that have been released. Some are proprietary and some are open source. So definitely a lot of people looking at this problem, and it's good to have like all those options. Another seminal paper that I found really, really interesting in the field is this paper from Rogerson at Gustave Roussy in France, where he basically did something that a lot of people thought a little bit crazy at the time, which is, can you predict a gene expression signature, in that case, CD8 T cell infiltration from radiology? And if you're successful at doing this, that's very beneficial because you don't need to do a biopsy, which is very invasive. and you don't have to do the sequencing, which could be very expensive, right? And so now you have this surrogate radiomics predictive a predictor for something that previously was only detectable using molecular profiles such as gene expression. And that worked surprisingly well. It happens that when the tumor is infiltrated by T cells, you really see an emerging phenotypes on radiology. So of course you don't really know which gene are overly or overexpressed or which pathways are activated, but you can definitely see a phenotype that's sufficient to build that surrogate biomarker. And they actually evaluated that in multiple trials, and a lot of other studies have kind of followed through that vein of that kind of direction, research direction. And basically now there is this concept of virtual biopsies, like what can you predict from the radiological images that usually previously were only possible to detect using molecular profiling. So people have tried to predict mutations, tumor mutation burden, all kind of immune signatures, inflammation, and whatnot. So it's really an active field of research. And again, the beauty here is that it's non-invasive. This kind of data are readily available in the clinic. There are many limitations. Radiomics is as complex as genomics data analysis in many ways. So you can make the same mistakes that everybody else is doing in terms of data leakage and properly validating your model, making sure that you're being transparent, et cetera, et cetera. But I think one of the kind of aspect that's very specific to radiomics is the kind of features and the kind of data you use to extract those features. And so there have been multiple papers to kind of understand what are the best approaches to properly document features, but also extract features that are relatively stable, so they're not fully dependent on scanners, for instance, or imaging protocols. And you can see here a series of like a review that explain a little bit the different initiatives that have been put. And I think the most important ones are probably the image biomarker standardization initiative, where they basically compute the features on phantom data, so the same kind of type of images across multiple centers and see what features are kind of are robust. And then there is the radiomics quality score that really try to kind of provide guidelines on how to best do your radiomics study, so you're not making all the obvious mistakes. Um, so I think the biggest challenge we've seen so far is that in radiomics there is this massive confounding factor, which is volume. And the reason is that we have all those kind of beautiful equations that extract radiomics features, but the truth is they're all very sensitive to volume. So if you have a small tumor, you can have a certain kind of range of radiomics features values, and if you have a large tumor, you can have a different range. And it could be counterintuitive. Like if you look at tumor intensity and tumor shape, you would think that it has nothing to do with volume, but because it's a big confounding factor, because a lot of those kind of features depends on the number of voxels in your region of interest. They're highly dependent on volume. And so if you look at this paper, for instance, we with actually the same author, with Hugo, we actually kind of went back to the original publication and decided to, and Mattia Welsh decided to do something quite interesting, which is let's randomize the voxel and see what happens to that signature. So that signature was like 4 radiomics features: intensity, shape, texture, wavelet, and it was predicting survival of lung cancer patients pretty well. And so what Mattia did was let's randomize the voxels in the image, and the signature should fail, right? If it really detects something intrinsic to the tumor based on the very specific arrangement and distribution of voxels, it should completely fail. The problem is that it did not. I would not say it got better, but it definitely got a similar prognostic value. And so it means that when you randomize the voxels, the only thing that you keep stable is the actual volume, the segmentation, the 3D segmentation of the tumor. But all the voxels in it are completely randomized, right? They shuffle from a sample from the whole images, and they're not really tumor specific anymore. And so the concern in the field was that despite all the sophistication of radiomics analysis, maybe we've just discovered that if you measure volume in a precise and robust way, which radiomics does, by the way, it just computes volume, that's all you need for prognostic value. That is true for lung cancer, head and neck cancer prognosis, but that is not true for many other cancer types, right? So I don't want to sound more dramatic than it is. There are definitely many tasks and many contexts where volume alone is purely predictive. But in the case of that seminal publication, there was definitely a very large confounding factor. And so um, people have tried to kind of build more sophisticated radiomics features. And here's an example with the Foundational Model for Cancer Imaging Biomarkers, or FMCIB, where basically they kind of took hundreds of thousands of radiological images, mainly CT and MRI, and basically use the self-supervised learning approach that is and transformers that is so characteristics of foundation models, and they build those new embedding space that could extract features that have not been engineered. We don't have a proper equation for them. We have a very complex, weighted formula which represent all those embedded features from the deep learning model. So that's exciting in principles because we go beyond those engineered features and hopefully we can extract a lot more new information in the images. But again, the question still remains: do we still recapitulate volume, or those features are intrinsically more informative? And actually, the same lab, Hugo again, developed a new version of that color vision foundation models, which we didn't really analyze yet. But you can see the activity in the field in terms of trying to go beyond those engineered features that are in PyRadiomics, LifeX, and other packages. Um, Another aspect of many studies is the fact that you really have to make data like kind of ready for the analysis. And DICOMs, which is the format where images are stored, are really rich data format. They can store a lot of metadata in many headers, which sometimes could be very tricky. So in imaging, you usually have the image of the tumor, but then you also have all the masks that represent the regions of interest. Could be the organs, could be the tumor, could be the lymph nodes. And you have to extract all that information. What Medimage Schultz is doing is kind of diving into all those metadata, which is basically this big house of raw medical imaging data, and try to understand the dependencies of the data. So if you have a segmentation or a mask, um, what's the baseline, the reference image for that mask, for instance? And you can see also RT-struct that are kind of segmentation used for radiation planning, for instance. Um, radiotherapy planning. So MediMeasure is kind of going into all the metadata and try to sort out all the dependencies so that you can make those data readily available for your analysis. And we've designed the package in a way that it really creates objects that could be directly used in PyTorch so that you can do your AI modeling much faster. And what Katie's got in the lab, who, by the way, she's the one who put all those slides together, so kudos to her. She basically thought deeply about these initial negative controls that Mattea Welch developed, and she tried to basically develop other um, negative controls that could be relevant for radiomics analysis. And here's the idea. So let's say you have this kind of round, perfectly round tumor. It's just a toy example. So you have the image, you have the region of interest, which is the circle, that's the tumor. And so everything outside the tumor is kind of the environment of the tumor, could be different organs and whatnot. And so you have this original image. So now you have different ways you can randomize the data. You can randomize the data from taking voxels from all over the place. You can basically shrink the tumor segmentation and try to simulate kind of how segmentation could vary if humans do different segmentations. And look at the kind of the ring outside the tumor, look at the ring inside the tumor. You can expand that segmentation, etc. So she kind of started by looking at the different variations across the segmentation itself. But then she decided to also investigate how you do the randomization. So um, you can see here this metric, a 3 by 3 matrix, where, for instance, shuffle and full means that you take the whole image and you just shuffling the existing voxels in that image. You can do the same within the region of interest. So that square is basically kind of the tumor, if you wish. And so now you keep the rest of the image intact, but you only shuffle the voxels within the region of interest. And then you do the exact opposite. You shuffle everything but the region of interest. You can do the same thing by not shuffling the existing voxel, but just taking generating random data from a uniform distribution. So you don't really respect the distribution of voxel intensities. Or you can do sampling. You just sample from either the full image or you actually sample from the distribution of voxels. So you kind of create synthetic data that way. And again, from either the full image, the region of interest, or everything but the region of interest. So suddenly those negative controls got quite a bit of more sophisticated. And then another aspect is that there are new AI tools that require kind of a bounding box around the tumor to extract those features. And that's the case for this foundation model for cancer imaging biomarker FMCIB. And so the question is, how do you build that bounding box? Do you make it fit perfectly around the tumor segmentation? Do you make it center and then you capture a little bit of what's around? That's the centroid. Or do you make it a cube? You kind of try to fit closely the tumor, but in a perfect cube. And those are very artificial decisions, but they're kind of software limitations. Like a lot of those foundation models, they actually require you to define this bounding box or this mask, right? So we also wanted to investigate the variability in those. And so here's an example, like that's not a toy example, that's a real example now. You have this tumor that's obviously not this perfect square, it has this weird shape. And now you can see the results of the randomization, all the negative controls. So at the top you have like random voxel generation from for the full images um, for everything but the region of interest and the region of interest and the different way you generate those voxel intensities. So now you can really generate a lot of different kind of random data to uh, to challenge your radiomics model. And so you basically now take those negative controls and you do exactly the same pipeline. You carry out the same pipelines than before. You develop your prognostic models, or you take your prognostic models and you use those data as input. And you can create basically imaging features for those random data. Um, we wanted to provide an easy way to share those data with the community because this is really computationally intensive to create those random data. So if we can uh, let people skip that whole step and leverage already the negative control directly, that would be beneficial. So we decided to design a new class in Python called radiomic set, which is kind of based on a multi-assay experiment structure. And now we have this bumpy matrix where each voxels are now have an additional dimension, which are all the negative controls. So now people can leverage those data. Obviously, the object is pretty heavy now because we have all those randomized data, but they're directly accessible for your analysis. And we put all that data into Orchestra, a platform where you can see all the code used to generate the resulting data object from the original data. So it's fully transparent and reproducible. And we wanted to evaluate kind of the effect of those randomizations, a little bit like what Mattea Welsh did in her initial paper. And so we decided to reuse this pipeline for head and neck cancer prognosis. But we had this brand new data set from Princess Margaret called RadCure, which is like 10 times bigger than everything else that has been published in the past. So it's like 3,500 patients. So it was kind of a great opportunity to really kind of mess up a little bit with the prognostic models that you can design based on the existing data set and really have this large external validation cohort. And the way we did this is we basically computed the correlation between features before and after randomization. So if you look at the feature-feature correlation, that's the first matrix on the left, you can see that kind of complex correlation, inter-feature correlation structure. And you can see that some features are very correlated with each other, especially if they're in the same class. So let's say a lot of shape features are highly correlated, but you can see that some are still intercorrelated. So you have this nice kind of complex structure. If everything goes well and you randomize the data, that correlation structure should be completely gone because it means they really depend on the specific arrangement of the voxels. And once you randomize, all those features don't make any sense and they're completely random. If the voxels actually don't matter, it's all about volume, it's all about the segmentation, then the correlation structure will be exactly the same, right? So those are the two extreme cases. And so we were wondering now with radiomics, for instance, where you would fit in terms of negative controls. And those are the results we obtained. So you can see there is some loss, especially when you randomize the full image, there is some loss in terms of correlation structure, but there are still features like shape that are perfectly correlated, right? So you're absolutely not, by randomizing the voxels, you're absolutely not changing the shape features, which kind of makes sense. It's really based on the segmentation, but you can see that a lot of structure is still conserved in the other radiomics type. So that's an indication that even when you do like the strongest negative control, which is randomize the whole image, you still conserve some structure, which is kind of worrisome. No, what is a lot more worrisome is that if like majority of the papers you claim that your radiomics features really depends on the tumor and you start randomizing the tumor, what's around the tumor, then you can see that that structure is really well conserved. So there is a bit that would be an indication that even though we like to believe that those radiomics features really depend on the voxels, they're probably really depending on something that is much more abstract, such as volume. And so we wanted to see whether it would affect the prognostic modeling. So we took those two papers, and ideally what you would see is that the original model has a very high concordance index, and once you start randomizing, that predictive value is completely gone. But that's absolutely not the case. You can see here that most of the data, the model using randomized data are equally prognostic. So again, an indication that you have this huge confounding factor. So we knew that based on Mattea Wells' study. Now we are investigating other tasks such as HPV predictions and so on, and see whether that would affect those tasks a lot more or a lot less, sorry. And I think that's going to be the case. I think for whatever reason, survival volume is so prognostic that it makes all the other features kind of completely useless. But I suspect that for other tasks, it's not going to be the case. So now we have the worst case scenario. Now we're going to investigate other tasks and see what happens. And it's a bit of a fact that radiomics is very volume driven. So the question is, do you see the same effect for FMCIB for this foundation model? And when you do the full, the correlation is completely gone. So that's good news. So FMCIB is definitely very specific to the actual arrangement of the voxels. But when you shuffle only the ROI, so what's inside the tumor, surprisingly, that correlation structure remains. So that was a big surprise for us. It basically means that FMCIB is deriving its embedding features mainly on non-ROI, so outside the tumor voxels, something that the original authors didn't know, right? So we came back to them and said, "Oh, that's really weird. Can we do more tests?" And now we're investigating this in more details. But according to our test, FMCIB is capturing what's around the tumor, not really what's inside the tumor, which is kind of interesting. And so next step, as I said, we want to apply that to this new tumor imaging batch, this new vision transformer that Hugo Art's lab is developing. And we're actually working with them to make sure that their model is not confounded by volume, and they can use some of the negative controls to actually fine-tune the model that way. We want to play a little bit more with the variation of the ROI, which I did not present today. And we want to include a lot more radiomics feature software such as MIRF and Pulsera to kind of have this toolbox that can investigate all the other radiomics feature software. And with that, I really would like to thank Katie, who's the main leader for the main lead for that project. Andrew Hope, who's my partner in crime for Radiomics, and who's also supervising Katie on this project, and the whole lab for the help. Thank you.